I'm David Nguyen, CEO of Brain Scanology, and in this presentation, we will provide proof of concept that we can detect malaria infection in microscopy without using Giemsa staining. According to UNICEF, in 2021, there were 247 million cases of malaria and 600,000 deaths. 77% of those deaths were in children under the age of 5. Because even though treatments for malaria exist, if you wait too long before diagnosing it, the body cannot recover. Thus, Brain Scanology wants to develop a machine learning algorithm that works through a smartphone camera that can be used to detect malaria in developing countries all around the world. I'd like to point out that there are single-use molecular test kits that make detecting malaria very easy. However, because they are single-use, this means they become very expensive over time for rural areas and developing nations. Therefore, Brain Scanology wants to develop a machine learning model that works with a smartphone, a glass slide, and a microscope attachment, because all of these three objects can be used many, many times. In microscopy, malaria is detected by staining a smear of red blood cells. The chemical is called Giemsa, and this chemical reveals the presence of parasites by making them look purplish. As you can see, these rings inside these red blood cells. This process, however, can take up to 2 days before you get the results. According to this article that suggests best practices in how to stain for malaria using Giemsa, they recommend that the blood smear be dried at least 1 hour to overnight, and that the staining step should last for about an hour. So this means it's at least 2 hours before the staining is done. And then the authors recommend that a specialist looks at 200 high-powered fields on this slide under a microscope before making conclusions. So our question is, how can we speed up this diagnostic process? Because if we can remove the need for Giemsa staining, then we can save money and shorten the wait time. If we automate the classification using machine learning and a smartphone camera, then we also shorten the wait time. We reduce the need for expertise to make a diagnosis, that is, even a medical assistant, as opposed to a licensed pathologist or hematologist, can make the diagnosis, meaning you can then serve more patients. But the challenge of diagnosing malaria without Giemsa staining is that you can no longer see the parasite. On the left, the parasites are in purple, but without Giemsa staining on the right, all you see are the red blood cells. So you have to measure something other than the purple parasites. The solution is to measure the shape of all cells in the slide. On the left are examples of uninfected red blood cells from somebody who has no malaria. And on the right are examples of cells that have the parasite, that is, they have a red asterisk. But most of these cells from this infected person are in the blue star category. That is, they are uninfected, but adjacent to infected cells. So the infected cells we refer to as malaria infected cells, or MICs, and the blue cells we refer to as non-infected adjacent cells, or NIAC cells. Again, these NIAC cells do not have the parasite, but they are floating around next to their neighbors who do have the parasite. And the majority of cells in a blood smear from somebody who has malaria consist of the blue stars, that is, the NIAC cells. So the question we asked is, are the shapes of these NIAC cells different than those in healthy individuals? And the way we measure that is to measure the outline of the cell, but we measure something called pure shape, which is very special to our software called Shape Genie. So if you measure objects at the original scale, you cannot separate the effect of size from the effect of shape. But Shape Genie allows us to resize these two objects such that they here have the same height. And when we resize them, we constrain what's called the aspect ratio, so the shape doesn't skew. And what we compare then is their shape apart from their size. We call this pure shape. And this is something only Shape Genie can do. We then apply what's called the LCPC transform through the radial grid system. For example, if this is the outline of our cell, then we find the midpoint of this object, and then we overlay a grid system. And this grid system allows us to find all intersections between the edge of the object and the radial lines. These dots then each have a distance to this red center. And this allows us to turn the shape in step 3 into the graph in step 4. We then apply the fast Fourier transform on the data in step 4 to get what's called the frequency plot in step 5. And this frequency plot is a static multidimensional representation of the shape in step 1. We say static because the numbers don't change, like your phone number. Your phone number consists of a series of numbers, but they're the same numbers every time somebody dials you, right? They don't change each time somebody calls you. Though this kind of data is complicated, it reveals hidden patterns in shapes that the human eye cannot see. And people have the option of creating a scalar value, that is a simple number, from this complicated multidimensional set of numbers. And one way to do it is to sum all these blue bars together to get one number. But in this presentation, we will work with the multidimensional data. For a brief description of the protocol, what we did was we segmented the outer shape of each cell, and then we found the longest internal line of each object. We rotated that line so that it's horizontal, and then we resized every object to be the same width, right? Notice here there's no white space between the dotted lines and the blue line. Okay, then we rotated these objects 90 degrees clockwise to get them vertical, and then we applied the radial grid processing system as described previously. Because we applied the radial grid system, which overlays 71 grid lines, typically the result is that each object has 71 rows of numbers. Think of this as each cell having a phone number that consists of 71 values. We then applied a method of dimension reduction known as principal component analysis on all of our cells to find the key differences that represent these groups of cells. Here is a three-dimensional plot of uninfected cells in green next to the NIAC cells of the vivax strain of malaria. So again, NIAC are the non-infected adjacent cells, right? They don't have the parasite, but they're swimming next to cells that do have the parasite. And these are from the strain of malaria called vivax. If there were no difference between healthy and NIAC cells, the blue and the green dots would be evenly distributed throughout this whole plot. However, the NIAC cells all gather within this window here, which we call the NIAC window. And here is a three-dimensional view of the same data showing that this window is restricted to a certain area of the 3D graph. We then did the same analysis on cells from a different strain of malaria. Instead of vivax, we then analyzed the falciparum strain of malaria. And here again, we found the same pattern. That is, the NIAC cells all occurred within the NIAC window, with the uninfected cells being scattered across both areas, some in the NIAC window and some outside of it. And these uninfected cells are from the NIH database, which is a gold standard of Giemsa staining for malaria infection. Though we were excited that we found the same pattern of this NIAC window in both the vivax strain and the falciparum strain, we realized that these cell images were from a pamphlet created by the Center for Disease and Control, or CDC, in the United States. So the CDC created this pamphlet with many images to teach people how to recognize Giemsa staining for malaria. And so before we wanted to get too excited about our findings, we wanted to see if we could validate this same pattern on data from other image sources. We knew that papers that studied malaria often contain images, right, of Giemsa staining. Therefore, we found these 7 papers that studied the falciparum strain and these 9 papers that studied the vivax strain and extracted the cell shapes from their figures. What we found was that NIAC cells from these 7 distinct papers exhibited the exact same pattern. That is, they all clustered here in this NIAC window. We found the exact same pattern from the 9 papers that studied the falciparum strain. All the NIAC cells, that is the non-infected adjacent cells, clustered in this NIAC window. And this was very pleasing to us because though none of our data is from a formal study, these papers were published across a period of 10 years from different places in the world, from different human subjects, different microscopes, and different staining parameters, yet they all exhibited the exact same pattern. That is, NIAC cells have quantitatively distinct features than healthy uninfected cells. And because of this, we believe that we are onto something, that there is a difference in shape between non-infected adjacent cells and healthy cells, even though both of these cells do not have the parasite in them. And in hindsight, this makes sense because these NIAC cells, though they don't have the parasite in them, they are swimming in the environment in which their infected neighbors are releasing chemicals, danger chemicals, telling their neighbors that something is wrong. And in this environment of stress hormones that are being released by their neighbors, they are likely in a mode of high caution. And therefore they adopt a shape that isn't what they normally look like. And so the next question is, how about the shape of malaria infected cells as opposed to their non-infected neighbors? Here we compared the shape of infected cells from the vivax strain and infected cells from the falciparum strain. And as you can see, they distribute relatively evenly. And interestingly, by themselves, they still exhibit the pattern of being in the NIAC window, right? So this tells you that this NIAC window is not an artifact of the uninfected cells, because when we remove the uninfected cells, the two infected groups still formed this NIAC window, telling you that there are quantitatively distinct features of cell shape due to malaria infection. Interestingly, if you look at this three-dimensional view of the same data, there does seem to be a plane that separates the green vivax infected cells from the blue falciparum infected cells, especially when they're outside of this NIAC window. And this might be evidence of a morphological difference between cells that are infected by vivax versus those infected by falciparum. But we need more data before we can reliably conclude this. When we pooled the malaria-infected cells from both strains together and then plotted them with the uninfected cells, we see that 96% of the infected cells are in the NIAC window, while only 4% of them are outside the NIAC window. This means that the vast majority of infected cells take on very distinct shapes that are different than a lot of cells from uninfected individuals. This fact then will guide us on what features we should include in our machine learning models that classify whether somebody is likely to be infected with malaria or not. So I'd like to point out limitations of this preliminary study. First and foremost, the source of our data are not from formal studies. We extracted cell shapes from images of publications. Our malaria-infected cells and our NIAC cells came from figures in published journal articles or a pamphlet from the CDC. Therefore, we need to validate what we've found from data that came from a formal study. And then we need to validate the patterns that we found in data sets from different studies. Lastly, we will also need to determine if we can find quantitative differences between malaria and babesiosis, because babesiosis can mimic malaria in Giemsa staining. As to how to build a classification model, there is more than one way. One option is to calculate percent of cells inside versus outside the NIAC window. Two is to classify the morphology of cells inside or outside the NIAC window. And three is a combination of the above along with other ideas. We need collaborators to send us blood smear data from different parts of the world so that we can build machine learning models that are specific to Africans, to Latin Americans, so on and so forth. We know that genetics and geography has a strong effect on how diseases manifest. Therefore, in order to build a machine learning model that works very well for Africans or for Latin Americans, we need data from those populations. If you're interested in partnering with us, we'd be happy to write grants together. Please reach out to me through email at dave@brainscanology.com. This is the end of our tutorial. We hope that Shape Genie helps you find amazing things in your data.